Right, so we're going to now look at one of the main hallmarks of cancer, which is sustained proliferative signaling. So sustained proliferative signaling, which basically is persistent activation of growth signaling. So again, as always, we like to draw our cells and denote the position of the nucleus, which contains our genetic material, and here is the cytoplasm where almost everything else is. Now, there are some examples of listed here that is going to help us in getting an overall understanding of what this sort of hallmark is all about. So, number one, we have EGFR, which actually stands for epidermal growth factor receptor, and don't mind the squiggle, but if you're going to look at that and see how it is regulated or rather dysregulated in the context of cancer. And first to mention that it is a receptor, so that it means it is going to be on the cell surface, so there can be more than one, of course. So some sort of receptor-like molecule. It is again implicated in different types of cancers, and is either overexpressed, so you would see many of them on the surface of the abnormal cell or abnormal cells compared to normal. So these can be found on normal cells, but they can be rather mutated. So for example, they all have this type of mutation that changes their receptor by a little bit, or that there are too many of them on the abnormal cell. And what these can do is send in signal to the cell from outside in signaling, right? So if something binds to this receptor, then we have activation of all these signaling pathways inside the cell. And actually two of these are listed here, the RAS MAPK and the PI3K AKT mTOR. So these two pathways actually exist within here. There are multiple different pathways as well, but each of these pathways have many proteins. So again, if we consider a receptor gets bound by its molecule, right? And then inside the cell we have a protein, and then a second protein, and a third protein, and so on and so forth, until we get some sort of response, a cellular response. And in the context of EGFR, this response is going to be growth signaling. Okay, so that's EGFR, and it is not only growth of the cell in it keeps on dividing, but as we had mentioned in our overview, that there can be formation of blood vessels that are going to supply. These are, for example, abnormal cells, and on the bottom we have normal cells. So if these abnormal cells have a lot of signaling going on inside, lots of binding, lots of signaling, then we can have formation of these blood vessels. So many more blood vessels are going to connect to the site of of the abnormal cells, and they're going to get lots of nutrients, oxygen, and whatever else they may need to begin dividing further on. Now, to continue on our conversation of these pathways, let's look at this number 2. Again, this number 3 and 4 are related to number 1, and perhaps these other pathways as well. But focusing on HER2, before we actually get there, is to mention that EGFR is equivalent to HER1. So there is HER1, which is equivalent to EGFR, and there is HER2, which is what we're going to focus on here next, and there are also HER3 and HER4, so on and so forth. But the main thing is that HER2, like HER1 or EGFR, increases proliferation, which is equivalent to cell division, or actually increased cell division, and in normal cases, when the cell is still normal, it has a role in differentiation, and it's a term that refers to the fact that say a very simple undifferentiated cell can become specialized cell. So I'm trying to make this very basic, but if we call it a very simple cell, so something very general, it has the potential to become, say cells that line your lungs, or cells that line your say stomach, or cells that line your, let's say, kidneys. So different types of cells can arise from one original cell, and HER2 in normal cases, in normal cells, is involved in differentiation. So, and these arrows refers to the process of differentiation. But in the abnormal cases, right, HER2 contributes to increased proliferation or cell division, as was the case with HER1 and EGFR. Now, HER2 is actually, it stands for, it did not mention that it stands for human epidermal growth factor receptor 2, but it is very more commonly mutated in breast and a gastric cancer. So breast and gastric cancer very much often have this HER2 mutation. Again, they either are overexpressed, so as in here we have too many of these on the cell surface, so overexpressed, or they have a mutation, right? So a little bit of a difference that makes this different from the normal, and these differences actually occur at the level of our DNA, and that translates to differences in the proteins that make up these receptors. So these receptors actually are protein. Now next, we're going to just touch on these last 3 pathways. So the WNT, or also called the Wnt pathway. Then we have the notch pathway, and then we have the hedgehog pathway. These three pathways you may or may not be recalling that are really important in the context of normal development. And of course, these are going to be then mutated. So mutated in cancer, right? So just to mention that as well, that we have many different types of signalings, and these are not even a conclusive list. Of course, they can range up to 100 if you, maybe, and can each have a dramatic influence on the behavior of these cells that are keep getting acquiring mutations in their DNA and translating them into dysregulated processes, such as increased signaling. Again, we had sustained proliferative signaling. Hopefully, this was able to give you an overview of potential topics you could further explore, and maybe we'll see you next